Hello everyone and welcome to my YouTube channel. So we were discussing about the different types of DNA extraction methods utilized in the laboratories for the isolation of DNA from bacterial cells, animal cells, viruses and plant cells. And the first method we had discussed was the phenol chloroform DNA extraction method, which is based on organic extraction. Okay. After that, we have also discussed that how can we precipitate the DNA and further quantify it. So all these topics we have covered in our previous videos along with different chemicals we utilize and the basic role of those chemicals. So for the basics of DNA extraction method, I would suggest you to go through those videos before coming through this one. Now today's topic is all about another DNA extraction method, that is the CTAB DNA extraction method, which we generally utilize for the isolation of DNA from plant cells. Now the question here arises is that why we require a different DNA extraction method for the isolation of DNA from plant cells. Why didn't we use the same method as we have utilized for bacterial cells or animal cells? Whatever chemicals we are using are same that we have used for bacterial cell DNA isolation or animal cell DNA isolation. But what is the difference here? That we have added certain chemicals in this method and we have also added certain steps. So why is it required? This why here is really important, and these kind of questions should strike your mind before you go through this topic. So the answer to all of these questions lies in the structure of the plant cell. Because we all know that the plant cell is much more rigid and complex as compared to bacterial cells or the animal cells. Animal cells, they don't have the cell wall, but bacterial cells, they have cell wall that we need to break so that we can get through our DNA. But the cell wall of the bacterial cell is not that much rigid and complex as compared to plant cells. We can easily break the cell wall of the bacterial cells by using certain chemicals or enzymes. But in case of plant cells, the cell wall is very rigid and very much complex, and it is very difficult for us to break that cell wall. And that is why we require different chemical methods and certain mechanical steps for the breakage of this cell wall. Okay, so the first reason is this. The second reason is the secondary metabolites that the plant produces for its different functions. Now we know that plants undergo certain metabolic pathways for their survival, like photosynthesis is there and other metabolic pathways are there. And during those metabolic pathways, they synthesize certain secondary metabolites. And these secondary metabolites, what they do, they will interfere with the DNA isolation. We will see how they will interfere later on. But for now, you should understand these two basic factors which are responsible for a different kind of DNA extraction method that we are utilizing for the isolation of DNA for plant cells. So the first one is the rigid structure of the cell wall and the second one is the secondary metabolites. Okay, so let's get started with the DNA extraction method for plants. We will read this topic in a stepwise manner and in a kind of flow chart, and we will be discussing the function of each and every chemical that we are using and what are the different steps we are including in this method. Okay, so let's begin with this topic. Okay. So we have discussed about the plant cell or the plant tissues. So the plant cell wall is very rigid. Why is it rigid? because of certain components present in it. And most of the cell wall is made up of polysaccharides. Celluloses, hemicelluloses are there. Certain other components are there like lignin, then your pectins. So all these components are making the cell wall very rigid, right? The second thing, during the metabolic pathways, they synthesize certain polyphenols, and one of them is tannin. Now, tannin is generally formed regularly in the plant cell, but when the plant cell undergoes certain damage, the amount of this tannin increases. It produces in a higher amount, and that is when we generally do the DNA isolation steps. This tannin, when it is released, it will hinder or it will interfere with this DNA isolation. Okay, so these two factors, the polysaccharides and the polyphenols, these two factors are playing the major role in the hindrance of the DNA isolation in plants, from plants basically. So we need to remove these two hindrances, and for that we require certain steps. Okay, so the first thing we need to do is to break the cell wall, to break these polysaccharides. And the second one is to remove the polyphenols. So we will see that how are we going to do this. So we have plant tissues. The first thing we need to do is to break the cell wall. How we are going to do that? By grinding the tissues. So we will get a mortar and pestle and we will grind these plant tissues. And when we'll grind them, we'll add certain amount of liquid nitrogen to it. Now, why we are adding this liquid nitrogen? Because of two important roles. This liquid nitrogen plays two important functions. The first one is it prevents the enzymatic reaction during the homogenization, and the second one is it helps in the formation of fine powder of plant tissues. Now, how is it going to happen? When we grind these plant tissues, what happens? The heat is generated. And because of this heat, the nucleases got synthesized and they activates. They come into function. And what is the role of the nucleases? Degradation of nucleic acid. So it may lead to the degradation of our DNA. We don't want this. So for that, we add liquid nitrogen to stabilize the temperature so that the temperature won't get higher. Right? So the first thing is it prevents the enzymatic reaction during the homogenization. Now when we add liquid nitrogen to our grinded tissues, the tissues become freezed, and when we grind further, a fine powder will be created of those tissues. So for creating a fine powder of plant tissues, we require the liquid nitrogen. So this is the role of liquid nitrogen here. Okay, now grinding is done. We got a powder form of our plant tissues. Okay, the cell wall is broken down somewhat, but still there are impurities and our DNA is not isolated. We want our DNA to be isolated. And during this homogenization, we are breaking the cell, right? We are damaging the plant tissues. So what it will do? For preventing this damage, it will release these polyphenols, tannins. Tannin, what it will do? It will lead to the prevention of that damage. Okay, so that is why this tannin will be synthesized. We don't want this tannin to be synthesized, because if it will be synthesized, it will bind with the DNA and it will not let it get purified or isolated. So to remove this tannin, we will use certain other chemicals in the CTAB extraction buffer that we are going to add in this grinded tissues. So when our tissues become grinded, what happens? In those grinded tissues, we will add the CTAB. the CTAB extraction buffer. And the CTAB extraction buffer contains certain chemicals. That is your CTAB, cetyl trimethyl ammonium bromide. Then you have certain salts, like you can use your sodium chloride. Then we have polyvinyl pyrrolidone. Then we have tris, EDTA, or we can use beta-mercaptoethanol or ascorbic acid, any one of it. Okay, so these are the chemicals we generally use in the CTAB extraction buffer. Now we will see that what is the function of all these chemicals that we are using. So first of all, in the CTAB extraction buffer, we have CTAB. So we have discussed that for the breaking down of the cell wall, we require three things. First one is the mechanical breakage, we have already done by grinding. Second one, we require certain chemicals. So the first chemical we are using is the CTAB. CTAB is a cationic detergent. What it will do, it will bind with the polysaccharides, it will make them permeable, and it will remove these polysaccharides, okay, by breaking them down. And when we use the CTAB along with different concentration of sodium chloride, the salt we are using, what this salt will do, this will lead to the precipitation of this polysaccharides. Okay, and we can easily remove these polysaccharides by centrifugation, because all these polysaccharides will come into the debris, and the supernatant we will have that contains your DNA. So we can remove that supernatant from the debris, and we can utilize the supernatant for further processing. So by utilizing the CTAB along with sodium chloride, the salt, we can easily precipitate the polysaccharides present in the cell wall. Okay, now this salt is also playing a very crucial role, because if we increase the amount of salt, then only these polysaccharides will be precipitated. They will become insoluble in water or in buffer, whatever we are using. But if the amount of salt is less, there is chance that your DNA get precipitated. Okay, so you need to see that whether your salt concentration is enough for the precipitation of these polysaccharides or not. So CTAB along with the salt will lead to the precipitation of polysaccharide. This is the use of the CTAB and NaCl in the CTAB extraction buffer. Now the second thing we are using here is polyvinyl pyrrolidone. So this chemical, why we are using this? Because of this polyphenols. What it will do? This PVP will bind with the tannin. And when it will bind with the tannin, tannin will not be free to bind with the DNA. And ultimately the DNA will be protected from getting degraded. So to protect the DNA from getting degraded by tannins, we require this PVP. Right? Next chemical we are using here is tris. In our previous lesson, we have seen that what is the purpose of this tris. This tris is working as a pH maintaining chemical. It maintains the pH of the solution. It maintains the pH of the reaction. Okay. Then we have EDTA. This EDTA is going to chelate the magnesium ions which are required for the DNAs, the enzyme which degrades the DNA. So when these magnesium ions will be chelated by the EDTA, they won't be available for DNAs because they are working as a cofactor. Okay, so DNAs require these cofactor for its function. So when magnesium ions will not be present in the solution, how will DNAs will work? Will be activated. So when there is no DNAs, there will be no degradation of DNA. Then we have beta-mercaptoethanol, or we can use ascorbic acid. So in this case, they are working here as the antioxidants. Okay, they are working here as antioxidants and they prevent the oxidation of these polyphenols so that they won't react with the DNA. Okay, so by preventing the oxidation of these polyphenols, they help in removal of these polyphenols from the solution and prevent the DNA from getting degraded. So this is the role of your CTAB extraction buffer. So when to our grinded tissues we have added the CTAB extraction buffer, all these things will be carried out. The degradation of the cell wall, then precipitation of polysaccharides, removal of your polyphenols, right? So all these things will be getting operated during that time. Now when we add the CTAB extraction buffer, we have mixed it well by vortexing it, and then what we will do, we will incubate this homogenate in water bath for 30 minutes at 60 degrees Celsius. So we will do the incubation. We got a homogenized mixture of the plant tissues. We will incubate in the water bath for 30 minutes at 60 degrees Celsius so that the remaining cell wall could degrade. Right? After this incubation, we will do the centrifugation step. What it will do, it will give you two things. First one is the supernatant and second one is the debris. This supernatant consists of the DNA and this debris here consists of all the unwanted particles, the cell wall, the remains of the cell wall, the degraded proteins, the degraded lipids, whatever decontaminants you don't want in your DNA, you will get them in the debris, the pellet. Okay, so you can remove this pellet easily by taking out the supernatant into the different fresh ependoff. So you will add the supernatant in a new ependoff, and what you will do, you will add certain amount of RNAs to it, because what RNAs will do, it will degrade the RNA. Okay, so that your DNA will be purified easily. It won't contain the RNA into it. So when we add the RNAs to it, we will again incubate that solution with at 32 degrees Celsius for 20 minutes. After that, we will add equal volume of chloroform isoamyl alcohol to it. Right? So remember the precipitation step now. We are moving towards it. So first of all, we will be adding the equal volume, that the amount we have of our supernatant, the equal volume should be added of your chloroform isoamyl alcohol in a ratio of 24:1. When we add the chloroform isoamyl alcohol and mix it well and again centrifuge it, we will get a supernatant and the your pellet. We don't require the pellet because it may contain the debris. We require the supernatant. We will transfer this supernatant in new ependoff and then we'll add chilled isopropanol to it. What the isopropanol will do, it will precipitate the DNA. We will incubate it at -20 degrees Celsius for 15 minutes. And after that, again, we will do the centrifugation step, right? And decant the supernatant without disturbing the pellet because we have already precipitated the DNA. If you want to know the function of isopropanol and how it is working, please go and watch the video of precipitation of DNA. Okay. After that, when you have your pellet there, what you will do, you will wash the pellet with 70 and 80% of alcohol. After washing, you will remove the ethanol and you will dry the pellet. Remember, the pellet should not be dried fully. right? You should just dry the pellet to an amount just to remove the excess amount of ethanol. Now when the pellet will be dried, you will dissolve this pellet in the TE buffer, that is your tris EDTA buffer, to maintain the DNA in a preserved form. So this is how you are going to extract a DNA from a plant cell. So all the steps I have covered here related to the plant DNA isolation are very crucial. Okay, that how you need to degrade the cell wall, how you need to remove the polysaccharides, how you need to remove the polyphenols, and ultimately how you are going to precipitate the DNA. So each and every step you should understand very carefully, and for further doubts you can comment me in the comment box, or you can also mail me at manishamalhotra92@gmail.com. And if you want the detailed notes on DNA purification, DNA isolation, DNA precipitation, quantification, and different methods, you can also mail me. And if you like the video, do like, share, and comment, and also subscribe to my channel if you are new. So thank you so much. Bye bye.